This is your second set of notes for cell communication, and we're going to pick up on this side of the big sheet where we left off before. So let's go ahead and zoom in. And we're going to start where we left off with tyrosine kinase receptors. So remember that this was a liver cell that was receiving an adrenaline ligand signal. So we're going to go ahead and stay with a liver cell and talk about insulin, because insulin will eventually cause the release of glucose from glycogen, and that's stored in the liver. So we'll stay in theme, and let's go ahead and I'll zoom in a little bit more. Let's review what insulin is really quick. Remember that insulin is a molecule that is made in, get my text box, and I can type this time, made in the pancreas, and this molecule is going to be released into the bloodstream, so we'll say that insulin is from the pancreas. When glucose levels are high, once you've eaten something and all the your small intestine has broken down, all the enzymes, in the small intestine have broken down all the carbs that you've eaten and absorbed into your small intestine and then into the bloodstream, and all that glucose needs to be brought into cells. So remember, insulin is the signaling molecule in charge of that. So from pancreas, when blood glucose is high, and in this case, we're actually going to need two insulin molecules. So what you're going to notice with this tyrosine kinase receptor, remember this is a kinase, so we're going to end up phosphorylating things. You actually need two glucoses. So here's our two tyrosine kinase receptors. We'll talk about what tyrosine is. It's when we look at the different amino acids, but you need two of these. These exist as a dimer. You don't need to know that term, but anything that kind of exists in pairs like that is called a dimer. So this is a tyrosine kinase receptor dimer, and there's two of them. And as is, they are inactive, but when they are bound to their ligands, remember these are signaling molecules, also called ligands, so I'll say two molecules of insulin. So insulin is the ligand in this case. They actually will connect to each other, so just kind of imagine them coming together. I'm just going to draw a little bridge here, but they physically will move together just a little bit because they're right next to each other in the cell membrane. And what's going to happen is a variety of responses is going to happen, and this is what's different from the G-linked receptor that we talked about last video. We're activating multiple pathways here, so that's one of the differences. One signal results in multiple responses. Remember, with our G protein receptor, one signal got one response, albeit a very fast and overwhelming response. With tyrosine kinase receptors, one signal gets you multiple responses. So a slight difference here. So all of these little buds are just tyrosines, which we'll see in later units what that is. But basically, these are going to get phosphorylated. So they all get phosphorylated, and they each are responsible, so same thing on the other side. They are each responsible for starting some kind of second messenger cascade, some kind of signal. So let's go ahead and look at three or four different ones. So one option, so I'll say option number one, is that you signal glut. Remember, glut is a vesicle filled with glut, which is a channel protein that allows glucose to facilitatively diffuse through it, where glut moves up into the membrane. Remember, we talked about this in a previous unit. Those glut channels get embedded into the cell membrane, and then glucose can diffuse from a high concentration in the blood to a lower concentration in the cell. So it diffuses into the cell, right? This is This is how glucose enters your cells in response to insulin. Right, so this is one pathway that can occur. This response, I didn't put it here, I forgot, is via a second messenger. So this molecule that I have here, the molecule that's responsible for stimulating the glut, the vesicle filled with glut, this molecule is a second messenger. Right, that is a second messenger. All of these are going to be governed by second messengers. And remember that second messengers can be cyclic AMP, and they can be calcium. I'll give you a third one in a minute. Right. A second reaction that could happen. Give me 1 second, my dog. Callie, out. Let's go. Good girl. All right. Actually, you know what? I'm going to go ahead and make a note of this. Let's go ahead and type this. So the first thing that could happen, oh, I hate when it doesn't let me type, is the translocation. Trans just means across, and location just means, you know, where you are, your location. So the movement, the translocation, the fancy way of saying it, of glut vesicles into the membrane. So one corresponds to outcome number one here via a second messenger, right? So let's look at number two. Number two is going to activate a different second messenger. We're going to get a variety of responses here, so each kind of response is going to be a little bit different. And let's say that this second messenger results in the phosphorylation. I think I can type down here actually. Can I? I'll learn one day. Or not. The phosphorylation of enzymes needed to make glycogen. Needed to make glycogen, right? If you're in a liver cell, this is where it would happen. Another one, so we'll do a third response, could be, here's our second messenger again. Our third response could be that you are basically doing the opposite here. You could end up with glycolysis. So actually in number two, we were doing dehydration synthesis, and in number three, we're doing hydrolysis. And let's just say that this were an adipose cell and a adipocyte, a fat cell. Maybe the second messenger would be in charge of metabolizing glucose, and I'll say in fat cells, metabolizing glucose into fatty acids. I'm just going to abbreviate that, fatty acids, to make triglycerides. Right, and all of these things, and again, if this were a fat cell, all of these things can happen simultaneously with a tyrosine kinase receptor, right? All four, well, I guess if this were a fat cell, four wouldn't happen in a liver cell also, but at least all three of these could happen in one liver cell at the same time from the same ligands, from the same signal, right? That's the difference between tyrosine kinase receptors, which are simultaneous, and your G protein receptors, which is just one response from one signal, albeit very fast and high magnitude. All right, so that's the difference between the two. The last Receptor that we're going to talk about here is an ion channel receptor. The default for an ion channel is to be closed, but if an ion binds, so if the ligand, remember this is a ligand again, binds to its ion channel, then it opens. So actually I'm just going to go ahead and say the channel opens and whatever the ion is can diffuse through. Right? This is diffusion. So if this were a sodium ion channel receptor and the ligand bound, then sodium would diffuse through. If it were potassium, potassium would diffuse through, and if it were calcium, calcium would diffuse through. Right, that's pretty straightforward. So the big picture here is that whichever of these three protein ligand channels you're using, this is all the reception stage, right, kind of up here on the top. All of this second messenger stuff, be it all these simultaneous pathways for the tyrosine kinase receptor, or just this one but very large and quick pathway for the G protein receptor, all of this is signal transduction. And then whatever the response is in this case, you've just liberated a bunch of glucose. Here you've metabolized, fatty acids, or you've liberated more glucoses, or maybe you've made glycogen, right? That's the response. Whatever the response is, you've added glut to the cell membrane. That's the response, right? So the last thing on this big sheet is the lipid ligand. The lipid ligand. So the lipid ligand, and let's stick with a specific example. Let's do testosterone. Both men and women have testosterone, it's just higher levels in men. Both men and women have estrogen, it's just higher levels in women. But again, this is coming through the blood. And just a reminder that testosterone is it's a cholesterol derivative, right? It's made from cholesterol. So I'll say cholesterol is the precursor to a molecule of testosterone, right? So it goes right through the membrane. It's hydrophobic, goes right through the hydrophobic center portion of the membrane, and it attaches to its cytoplasmic receptor, right? So it attaches to its receptor, and the receptor testosterone, or the receptor ligand combo, is going to diffuse. Remember, these molecules are just diffusing all around the cell. So it's going to diffuse to the nucleus. Testosterone acts on the nucleus. And this complex becomes what is called a transcription factor. And we'll talk about this more later, but transcription is when you go from DNA to messenger RNA. Right? So what this does is basically turn the genes on. Here's your double helix that I have drawn here. It turns genes on. Turns on genes, right? And if this is testosterone, maybe the transcription factor attaches here. Maybe this particular gene is involved in making a lot of hair, right? Armpit hair, facial hair, chest hair, and that gene gets turned on, right? This is what happens in puberty, and if you're a middle school boy. And maybe it attaches to this gene right here. Maybe that is involved in increased muscle mass. Literally more muscle cells as a result of testosterone turning on genes. Maybe it involves lowering your voice via a longer or larger larynx. So let's say enlarged / elongated larynx. It's your vocal box, right? So that, think of a stringed instrument. Short strings have a higher pitch, longer strings like a bass, or a bass is a good instrument that has long strings. It has a very deep kind of resonating sound to it, whereas a violin might have a higher sound, right? So again, the whole point is that these lipid hormone signals, remember these are hormones, testosterone is a hormone, can diffuse directly through the membrane, attach to its receptor. In this case, testosterone acts on the nucleus, and depending on whatever genes it activates, it can change muscle mass, the amount of hair, and other factors. Estrogen works the same way. This is just one specific example, but for estrogen, it's the effects are different. You have breast development and fat tissue gets added. So the effects might be different depending on what the ligand is, but this is again just one example of what could happen, just so that you have something concrete to work with rather than generalized concepts. Right. So on the back, I think we're good with this side. I'm just looking over to make sure I didn't miss anything. I think we're good to go. Let's go ahead and flip over to the back. Now my big sheet has extra stuff on it, so ignore all of this. We're not doing that this year. There's a nice overview here on the left, but I do want to draw your attention to the second messengers, because we did add a third messenger, or I told you I was going to. I just haven't done it yet. Go ahead and add IP3. I just want you to be aware of these second messengers, and you don't really have to know what IP3 means. I mean, I can tell you. I'll tell you. It's another inositol triphosphate. It's another second messenger, but whenever you see these, I just want you to say, oh, I recognize that as a second messenger. It's going to be involved in a phosphorylation or signal cascade in some kind of cell response. That's all I want you to take away from these second messengers. I want you to see them. Boom, that's a second messenger. All right. So let's look at how some drugs work. Let's talk about drugs. So in your in your body, kind of without drugs, you have natural proteins and other, most of the time they're proteins, but substances that bind to receptors that produce some kind of cell activity, just like we've been talking about, right? These substances have specific shapes that fit into the active site of these receptors, right? And the shape matters. They have to match. These are substrate-specific receptors. It's not just any old substrate. They have to match, right? If you have a drug that increases a cellular response, it's called an agonist drug. Agonist drugs have the same basic shape, at least where they contact the receptor. So here's kind of the normal body molecule, the square, and the pentagon has the same basic kind of shape match, even though the rest of it might not. And that's actually the main way that drugs work. When you take any kind of drug, it's the shape of the drug that is what actually ends up mattering. And in this case, because you're adding all of these molecules with the same shape, that is going to increase cell activity. Other drugs reduce cell response. These are called antagonist drugs. So they don't have kind of a perfect shape match, but they have a good enough shape match to get in the way. So I've drawn this kind of as a little loop here, and it blocks the receptor. And the result is that whatever cell response is produced gets halted, right? It gets stopped. So this response actually blocks, where this drug blocks the receptor and stops the response. Right. So let's just make a note of this. Right above, let's say for drugs, most drugs interfere with cell signaling to disturb or fix a problem, either as an agonist drug or an antagonist drug. All right, so the last thing here, and then we're done, is apoptosis. We're going to talk about how this actually ends up working with cell signals. So remember, apoptosis is programmed cell death. So let's see how it works. Here's a cell, and here's a mitochondria, and every cell in your body has a receptor for death signaling molecule. A big clunky name, but there's a receptor that is waiting for basically a death signal. And if there's no death signal, then nothing is wrong and the cell just lives its happy, normal cell life. But on the mitochondria, you have proteins called SED9. And proteins inhibit SED4. That's what they do. They inhibit actually SED4 and SED3. So think of SED9 as a bully, right? it's preventing SED4 and SED3 from doing their jobs. And we denote this by using a line with like a little line at the end and then a negative sign. So this means that this protein, SED9, is inhibiting, it's blocking the action of SED4 and blocking the action of SED3. So in a typical, normal, healthy, happy cell, SED9 is active and SEDS3 and SEDS4 are inactive. Unless something goes wrong, right? Maybe you fail to pass a checkpoint, and you're slated for apoptosis, right? So a signaling death molecule is going to bind to the receptor for signaling death molecule, and that receptor molecule will inhibit SED9. So now SED9 becomes inactive. Well, if SED9 is inactive, it can't inhibit SEDS4 or SED3, so SED4 or SED3 become active. And when they become active, they activate proteases, which break down proteins, and nucleases, which break down DNA. And this is what causes cells to apoptose. This is why cells go through apoptosis. This ends up in the cell forming these blebs, remember these little pouches of cytoplasm and nutrients and materials, and the cell just kind of blebs to death, right? It just buds off of itself until it just doesn't anymore, until it doesn't exist. So we can make a little note that this is when blebs are when the cell is digested from the inside. And just as a reminder, the blebs are not thrown out, they are recycled, reused by adjacent cells. Right. So go back through these set of notes, try to explain how drugs work, how apoptosis works, but on this side of the big sheet, really focus on kind of the big picture, knowing that or knowing how these receptors work, be it a G protein linked receptor, a tyrosine kinase receptor, or an ion channel receptor, right? With your G protein linked receptor, one signal gets you one overwhelming and quick response. With tyrosine kinase receptor, one signal gets you a variety, multiple responses, right? If the signal is a protein signal, it has to go through one of these receptors. It can't go through the membrane. But if you have a lipid signal, like testosterone, it just diffuses right on through, no problem. Binds to a cytoplasmic receptor, which can then, in a lot of cases, not all of them, but in most cases, it'll act as some kind of transcription factor that regulates DNA activity. Right. So I hope this helped. We'll work on this in class. We'll work on the explanations of it, and I will see you next time.